Hello guys, welcome you all to Unacademy. And today we are going to, myself Himanshu, and today we are going to start molecules and their interaction relevant to the biology, which is the first module in case of CSIR NET Life Science Examination. So are you all online present? Please comment on the comment box and let me know that you are all present. So I welcome you all. Please guys, all of you, those who are online, they can post their messages, say me hi, so that I can know that people are here online. I welcome you all to this session on Unacademy. So let's begin today's session. We'll be studying molecules and their interaction relevant to biology. This is an important module as far as CSIR NET Life Science Examination is concerned. And we are going to start this module. So let's begin. Before we start, I would like to tell that each one of you needs to install Unacademy Learning app, because this is a beautiful platform where you will be getting your doubts cleared. You can study from various educators. For that you need to go to Google Play Store. You need to go to Unacademy Learning app, where you need to install this app, and you need to select your goal. In the select your goal, you need to select CSIR UGC NET. Once you do that, you will be getting access to various educators courses online, and also you can opt for the paid subscription. Now this is important because once you subscribe, you will be getting exclusive guidance, exclusive membership, where once you buy, you will be able to select the courses which you want to study at your own pace, at your own comfort. So this subscription of CSIR UGC NET in Unacademy is available for 1 month, 3 months, 6 months, 12 months and 24 months. I would suggest each one of you to get into 12 months and 24 months of subscription, and you can enter my code HY10. With that you will get a 10% off, and the entire course can be bought for 10,395. You can do the payment either by card or by EMI or by net banking. So before we start, I'd like to also tell that we are running free classes on YouTube, and they are also very comprehensive. You can attend those classes also, but if you want exclusive guidance, you should need to subscribe the Unacademy subscription on the Unacademy app, learning app. I told you the procedure for that. So let's begin today's class. So we all know about elements and compound, that matter is made up of elements. That matter is made up of elements. An element is a substance that cannot be broken down to other substances by chemical reaction. That cannot be broken down. So that is a basic criteria for being an element. Now what is a compound? A compound is a substance that consists of two or more elements in a fixed ratio. So fixed ratio is important, and compound has characteristics different from those of its elements. So this is something basic about elements and compounds. Now as we know about subatomic particles, that atoms are composed of subatomic particles. Atoms are composed of subatomic particles, namely neutrons, protons and electrons. Electrons are negatively charged, protons are positive in nature, and neutrons have no electrical charge, they are neutral. Neutrons and protons form the atomic nucleus. So N plus P, atomic nucleus, and electrons form a cloud around the nucleus. this is the basic introduction which I am giving before starting this today's lecture. And neutron mass and proton mass are almost identical and are measured in daltons. So what is atomic number and atomic mass? Anybody can explain it? So atoms of various elements differ in the number of subatomic particles. An atom's atomic number is the number of protons in the nucleus. An atom's mass number is sum of protons plus neutrons in the nucleus. Atomic mass is the atom's total mass, which can be approximated by the mass number. Isotopes. What are isotopes? That all atoms of an element have same number of protons but may differ in the number of neutrons. So isotypes of two atoms of an element that differ in the number of neutrons, that are isotopes. And what are radioactive isotopes? That decays, those that decay spontaneously, giving off particles and energy, are characterized into radioactive isotopes. So some of the applications of radioactive isotopes in biological research, it starts from dating fossils, tracing atoms through metabolic process, diagnosing medical disorders. So these are all the applications. Next we come to valence electron. They are those electrons which are found in the outermost shell. So these are those electrons which are found in the outermost shell or the valence shell. The chemical behavior of an atom is mostly determined by the valence electrons. And elements with full valence shell are chemically inert. So what is an orbital? Electron orbital is a three-dimensional space. It is a three-dimensional space where an electron is found 90% of the time. Each electron shell consists of a specific number of orbitals. So this is all about electron orbitals. Now next we move on to what are non-polar covalent bonds and polar covalent bonds. In non-polar covalent bonds, the atoms share the electron equally. In polar covalent bond, one atom is more electronegative and the atoms do not share the electrons equally. Unequal sharing of electrons causes a partial positive or negative charge from each atom or molecule. What are ionic bonds? Atoms sometimes strip electrons from their bonding partner. An example is the transfer of electron from sodium to chlorine. So here the transfer of an electron takes place and both the atoms have charges. The charged atom is called as ion. Now then comes the hydrogen bond. It is formed when a hydrogen atom covalently bonded to one electronegative atom is also attracted to another electronegative atom. In living cells, the electronegative partners are usually oxygen and nitrogen atoms. So hydrogen bond is important. Next we come to van der Waals interaction. So if electrons are distributed asymmetrically in molecules or atoms, they can result in hotspot of positive or negative charges. So van der Waals interactions are the interaction between molecules that are close together as the result of these charges. So the biological macromolecules have well-defined stoichiometry and geometry. They have well-defined stoichiometry and geometry. They are not readily broken into tiny pieces. So monomer is the building block. Next we come to biomolecules, biological macromolecules as I say. So from the amino acids, the proteins are formed. From the nucleic acid, the DNA or RNA are formed. The primary structure is one-dimensional sequence. Secondary structure is local arrangement such as the alpha helices, beta sheets and turns. Supersecondary structure consists of hairpins, corners, alpha, beta, alpha motors. Tertiary structure consists of 3D structure that is stabilized by hydrogen bond, hydrophobic forces, van der Waal, and charges. And the quaternary structure consists of arrangement of subunits, for example, in case of hemoglobin. Now there is a difference between configuration and conformation. Can anybody tell me what is the difference between conformation and configuration? Can anybody type, those attending the session? Yes, everyone. So configuration is defined by the chemical, in case of covalent bond, must break the bond to change the configuration. Example, L amino acids and D amino acid. So that is confirmation. Configuration, sorry. Conformation is the spatial arrangement. For example, an amino acid polymer can have a huge number of different conformations, one of which is natively folded protein. Now next we come to central dogma of molecular biology. So as we know, there is DNA, and DNA undergoes transcription. DNA undergoes transcription with the help of RNA polymerase to form mRNA, and mRNA undergoes translation with the help of ribosomes to form the protein, and DNA polymerase undergoes replication to form genomic DNA. So from DNA to mRNA, from mRNA to protein, it is a central dogma of molecular biology, and it describes how the genetic information encoded in the letter sequence of the DNA is first transcribed and then translated into amino acid sequence into the proteins. So this is a central dogma of the molecular biology. And proteins ultimately encode for enzyme. Enzymes catalyze the reaction in the organism, and proteins consist of the building blocks of the organism. Now this is the central dogma in one picture. So in case of a prokaryotic cell and the eukaryotic cell, so in the prokaryotic cell, everything goes inside a cell because it does not have a well-defined nucleus, whereas a eukaryotic cell has a well-defined nucleus. So transcription and post-transcription into mRNA takes place in the nucleus, and translation takes place in the outside the nuclear membrane, outside the nucleus, where ribosomes are present. So post-translation, protein folds up and protein active, and the active protein is obtained, which encodes for a particular function. Whereas in case of prokaryotic cell, transcription and translation go side by side. So this is how central dogma looks in a prokaryotic, that is a bacterial cell, and a eukaryotic, that is a higher cell. So this is the transcription process, whereby the RNA polymerase binds at a promoter and unwinds the DNA and starts the synthesis of an RNA copy of a gene. So this is the unwinding process that is going on. This is the structure of the DNA and the RNA. This one is the DNA. There are 4 monomers building blocks. RNA has ribose instead of 2' deoxyribose. RNA has uridine instead of thymidine. So ribonucleic acids and the 2' deoxyribonucleic acid are the polymers that are constructed from the nucleotide monomers. The nucleotides are distinguished by the nucleobases, which can either be pyrimidines or purine types. The polynucleotide sequence is a chain that extends from 5' to the 3' terminus. So this is the case of pyrimidines, and this is the case of purines, and this is known as a nucleobase. Together it's known as the nucleoside, and if the phosphate group is included, it's known as a nucleotide. So what are the stabilizing factors in the double-stranded DNA? So once the bonding takes place, the hydrogen atom is very important. Hydrogen bond is very important. It plays a very important role, hydrogen bond. So between the anti-parallel and the parallel strands of the DNA, the hydrogen bond is there, and it controls the bond, the stability of the double-stranded DNA. Now this is a bit of nucleic acid nomenclature. When there is adenine, so base plus ribose sugar nucleoside is known as adenosine. Base plus deoxyribose sugar is known as deoxynucleoside, that is deoxyadenosine in this case. And the ribose sugar plus the phosphate is known as the adenosine monophosphate. This is cytosine. This is the same for guanine. This is the same for thymine. Same for uracil. So this is a nomenclature of the nucleic acids. We are having three types of DNA molecule, namely bDNA, aDNA, and zDNA. Now, this is the double helix structure of the DNA molecule. This is the double helix structure of the DNA molecule. This is the minor group, and this one is the major group. So major group and minor group are there, and bDNA is right-handed. It is the most common form. It has 10.5 base pairs per ton. It has 3.4 nanometer of pitch and 3.34 nanometer of rise, and it is adopted in aqueous. Whereas in case of aDNA is also right-handed, but it is broader than bDNA. As you can see, it is much broader. It is much broader than bDNA. Compared to aDNA, it is much broader. So this is the aDNA, and this is the bDNA, and this is the aDNA. So aDNA is much broader as compared to the bDNA. It has 0.26 nanometer rise. It has 11 base pair plus per turn. It has 2.8 nanometer pitch, and it is adopted in non-aqueous. Most common form for RNA, has a hole down the center. And this is the zDNA. zDNA is left-handed. It is zigzaggy, and it has 12 base pair per turn, and it is adopted sometimes by the CG repeats. This is also one of the form. This is a cruciform form of DNA, and this is the triple strand form of the DNA or helix. So this is the basic difference between the aDNA, bDNA, and the zDNA. So this is an important concept, and each year several questions come from this topic, and you need to be well versed in this topic. This is the aDNA, this is the bDNA, and this is the zDNA. So this is the structure of the DNA and the RNA. Then there is a concept of linking number, twist, and right. So L is the linking number, and it is constituted from the T plus W. T is the twist and W is the right. So linking number is the number of times one edge of a ribbon linked around the other. It is a topological property. It cannot change without cutting. And T is the twist that contains the winding of the Watson-Crick integrated angle of twist by 2 pi along the length. It is not an integer necessarily, and W is the width, that is the wrapping of the ribbon axis around itself. It is non-integer and geometric property. And sigma is the superhelical density. It is calculated by W by T, width by twist. It is known as sigma. So this is the central dogma once again continued. So as you know that translation takes place inside the ribosome with the help of the tRNA. and this is the start codon AUG. This is the start codon. This is the start codon. This is the mRNA. And this is the direction of reading of codons in the translation, and this is the stop codon. UAA is the stop codon. So start codon and stop codon are there. So once the translation starts, it starts from the start codon and it ends at a stop codon, making a peptide using mRNA as the coding template. This is a diagram about the genetic code, how the genetic code looks. So a triplet of code for a particular amino acids, like U and UAC codes for tyrosine. There are 64 codons group. Next we come to the structure of proteins. Now structure of proteins, the primary structure is the amino acid sequence. So there are 20 amino acids common to all organisms, and each amino group, carboxyl group, R group and hydrogen in the tetrahedral symmetry. Almost all the organisms have L chirality, but some viruses have mirror image D chirality. They are linked together by the peptide bond. Peptide bond can be trans or cis, and proteins have prosthetic groups and amino acids can get modified. So there are two important angles. So one is the phi angle, that is between N and nitrogen and C alpha. So this is the phi angle, and this is the psi angle between C alpha C bond. So Ramachandran plot is there. It shows allowed angles. Actually the disallowed angle due to the steric hindrance. So this is the Ramachandran plot. There is the psi and the phi values. So this is the psi values and this is the phi values. So this is the right-handed alpha helix, this is the beta sheet, and this is left-handed alpha helix. So these are the permissible values of the allowed angles. So phi is between N and C alpha bond, and psi is between C alpha bond. So primary structure of the protein consists of the amino acid sequences. So there are 20 raised to the power n different sequences, but the converse is not true. So this is a little bit higher stuff. We will be seeing at a later stage. So from here, if we know that the Gibbs free energy is positive, it is hydrophobic in nature. If it is negative, it is hydrophilic in nature. And proteins have net charge and pocket of positive and negative values on themselves. So isoelectric point is a pH where the net charge on the protein is 0. Now again I would like to remind you all that please you can go and install the Unacademy learning app on the Google Play. You will need to select your goal where you will be getting CSIR UGC NET, and once you enter your goal, you will be seeing that there are courses from various educators. These are the live courses as well as the recorded courses. You can get the subscription where you will get the exclusive guidance. The subscription is for 12 months, 24 months, 1 month, 3 months and 6 months, but I would suggest go for 12 months and 24 months. You can use my referral code HY10. By entering this code, you will get a discount of 10%. So the entire course for 12 months you can buy for 10,395 instead of 11,550. You can select a payment method either by card or by EMI or by net banking, so you can get your subscription done. And there will be classes going on like the one that is going on now on YouTube, where you will be getting daily guidance as per the class schedule. So these are the free classes and will be available all the time. And if you get for the Unacademy CSIR UGC NET subscription, you will get exclusive guidance where you can get hold of the classes from various educators on this platform. So I would like to मैं आप लोगों से गुजारिश करना चाहूँगा कि आप लोग CSIR UGC नेट सब्सक्रिप्शन में एनरोल करें और क्लासेस का एक्सक्लूसिव गाइडेंस का फ़ायदा उठाएं. So let's go on. Now the structure of the proteins, as we saw, the primary structure is the amino acid sequence. So there are negatively charged amino acids, there are positively charged amino acids, there are hydrophobic amino acids which consist of non-polar, polar. There are polar amino acids which are uncharged also. So these are the different types of amino acids which are negatively charged, namely the aspartate and the glutamate. The positive charge are the lysine, arginine and the histidine. There are total 20 different amino acids. There are 20 different amino acids, and this one is the negatively charged amino acid. This one is the positively charged amino acid. And there are 20 different amino acids. This one is the non-polar amino acids. Non-polar which are hydrophobic, including tryptophan and phenylalanine. Non-polar hydrophobic amino acids which are alkyl. Tryptophan and phenylalanine are aromatic, and alanine, valine, leucine, isoleucine, proline are alkyl amino acids which are non-polar hydrophobic, and non-polar hydrophobic consist of methionine. Polar amino acid consists of glycine, threonine and serine. Polar amino acid with the disulfide adjacent consists of cysteine. Amines in case of amino acid consist of asparagine and glutamine. And polar amino acids in case of aromatic contains tyrosine. Now this is the secondary structure, that is the alpha helix, and alpha helix is the right-handed helix. It is a right-handed helix. It has 0.15 nanometer translation rise. It has 100-degree rotation twist. It has 3.6 residues per turn. It has a pitch of 0.54 nanometers, and it is stabilized by hydrogen bonds between the NH and the CO group. NH and the CO group. So this is the hydrogen bond. Between the CO of the nth and NH of the N+4 residue. This is also a secondary structure. This is the beta strand. It is parallel or anti-parallel in nature. Distance between the residues is 0.35 nanometer. Hydrogen bond between NH and CO groups of adjacent strand stabilized structure. This is the hydro order structure, which consists of beta turn, beta hairpin, Greek key, alpha and beta barrel. Hydrogen bonding disfavored in aqueous environment as beta sheets inside the globular proteins. This is the third tertiary structure, which is the overall 3D structure. And next comes the quaternary structure. For example, in case of hemoglobin, there is a difference between homodimer and heterodimer. Hemoglobin is a heterotetramer. There are various forces, as I told in beginning, uh, such as the London forces and van der Waal interaction, induced dipole interaction, hydrogen bonds. So these are all the hydrogen bonds. Now pairing is of two types. One is the Watson-Crick pairing and other one is the non-Watson-Crick base pairing. Next we move to the thermodynamics. So thermodynamics, in this section we are asking that in which direction a biochemical reaction will proceed. So this depends on two quantities. One is the enthalpy, other one is the entropy. Enthalpy is given by delta H and entropy is given by delta S. Enthalpy is correspond to the sum of all the potential energies as we discussed in the introduction, and entropy is related to the number of ways the system can adopt. In other words, it depends on the Gibbs free energy of the reaction. So this is our equation of Gibbs free energy calculation where delta G naught is equal to delta H naught minus T delta S naught. So this is the enthalpy. Delta H naught is the enthalpy. This is the entropy. Delta S naught. T is the temperature and delta G naught is the Gibbs free energy. So the Gibbs free energy of the reaction will determine whether the reaction will proceed further or the reaction will proceed backward. So how is enthalpy useful? So enthalpy, uh, the change between the initial and the final state of a biochemical process, it is easily measured as the heat that is generated or absorbed. So measuring the enthalpy of oxidation of glucose to CO2 and water directly in muscles would be difficult. But since the enthalpy is a state function, we can measure the enthalpy of the reaction in any apparatus and get the same result. This is again the Gibbs. So if the delta G for a particular process is negative, that process is spontaneous because it leads in the direction of equilibrium. And energy minimization and entropy maximization play an important part in determining the position of the equilibrium. There is one more equation for calculating the Gibbs free energy, which is given by delta G is equals to minus RT ln K equilibrium. We'll be seeing this slide again. So Gibbs free energy in real life, for real life biochemical reaction, we need to consider delta G for the object under study, for example, the protein or the reaction, and delta G for the solvent, usually water. The water molecule form hydrogen bonds, and fixing the water molecules will cause decrease entropy of the system. So delta G total is given by the delta G of the reactants plus delta G of water. So any doubts from anyone so far? Good evening, Shraddha. I welcome you to this platform. If you are able to hear me, guys, then please post your say hi. Now, what is the application of gives free energy to protein and DNA stability? We need to consider enthalpy part of the protein, and there are potential energies we discussed: bonding potential, hydrogen bonds, charge-charge interaction, dipole-dipole interaction, and van der Waal interactions. So we need to consider the entropy part of the protein or the DNA. Folded protein has low entropy and unfolded protein has high entropy. We need to consider the enthalpy of water. Water forms hydrogen bonds. We need to consider the entropy of water. We need to consider enthalpy of ions in the solution, that is the charge by charge interaction, and we need to consider the entropy of ions in the solution. So this is the application of gives free energy to the protein and DNA stability. Perhaps most important contribution to protein folding is the hydrophobic effect. The hydrophobic substances do not want to touch water, and it is perhaps the most important contribution to protein folding. Hydrophobic residues are on the protein inside, bases are on the DNAs inside and don't contribute much. So this is the hydrophobic effect. It is known as the entropy effect. It is the transfer of hydrophobic residues from water to non-polar solvent, example benzene. Often a small delta H but a large favorable delta S component. Why? Because sticking a hydrophobic substance into water makes water form a fixed cavity around the substance. This cost decreases the entropy of the water. So guys, if you have any doubt, you can post your queries so far. If you have any confusion, once again I would like to tell that you need to install the Unacademy Learning app, and you can go to the Google Play Store, whereby you will be finding this Unacademy Learning app. You need to install this app and you need to select your goal. Once you select your goal, you will be getting into CSIR UGC NET category, and you will be getting access to various educator classes. There are free classes as well as there are paid subscription. So paid subscription ensures that you get the entire access to the course. So there is subscription for 1 month, 3 months, 6 months, 12 months and 24 months. I would suggest that you go for the 12 months or 24 months subscription. You can use my code HY10 referral code. You will be getting a discount of 10%, and you need to proceed to the payment for confirming the details. So you can do the payment either by the credit card or the debit card or the EMI or by the net banking. So by doing this, you will be able to get the access to the course at your own doorsteps, और आप अपने ही कंफर्ट पे, अपने ही समय पे, जब आप चाहें, जितने बजे भी आप चाहें, जितने वक्त भी आप चाहें, जितनी बार चाहें, आप कोर्सेज को देख सकते हैं. आपको नोट्स मिलते हैं वहाँ पे, आपको मॉक टेस्ट मिलता है, आपको क्विजेस मिलते हैं, आपको एक्सक्लूसिव गाइडेंस मिलती है. So this is all what is very less when compared to a conventional coaching, because when you will be getting when you will be going to attend classes, it will easily cost around 40,000 to 50,000 plus other charges. So compared to that, and for this longer duration of time, it is an excellent opportunity for each and everyone to get access to the classes. So please get hold of these classes as soon as possible. Our courses are running live, and you can enjoy the learning experience here on Unacademy. We are also holding classes on YouTube. and these are the free live classes like the one that is going on right now, and you need to be very attentive because these are also the important classes and a lot of important concepts will be discussed here also. So guys, please uh, focus, and you can post your queries, any queries you are all are having. If you are having any queries, then please post in the comment box. Yes guys, please post your queries in the comment box. Yes guys, any queries, any doubt you are having, please post your queries or doubt. So I'd like to remind at what all we studied today. So in this session, so let me remind at what all we studied today. So we studied about the atomic number and the mass number. We studied about the just a second. We studied about the elements and the compounds. We studied about the subatomic particles that are the neutrons, protons, and the electrons. We studied about the atomic number and the atomic mass. We studied about the isotopes. We studied about the electron orbitals. We studied about the ionic bonds. We studied about the hydrogen bonds, van der Waal interaction. We studied about the central dogma of molecular biology, how the DNA is transcribed into mRNA and mRNA is translated into protein, and protein encodes for enzyme that catalyze reaction in organisms. So this is the central dogma. In the prokaryotic versus eukaryotic cell we studied, and this is how the transcription begins. We studied the structure of the DNA and the RNA. We studied what are the stabilizing factors in the double-stranded DNA. We studied a bit about nucleic acid nomenclature. Then we moved on to the three different types of DNA, basically the bDNA, aDNA, and the zDNA, where we studied their structure, how they are classified. Then we move to the linking number, twist, and the ride, and how they are calculated and what is their importance. This is the central dogma that was continued again. Over here, translation was shown. We also depicted the start codon and the stop codon in this case. This is genetic code, which is same in all the organisms. Then we came to the structure of protein, where we studied the primary structure, including the Ramachandran plot, and we studied about the primary structure. We studied about the individual amino acids that were negatively charged, positively charged, non-polar hydrophobic, aromatic, as well as non-polar hydrophobic amino acid that were alkyl in nature. And we also studied about the polar amino acids, and we also studied about the structure of the proteins, the secondary structure, which was the alpha helix in this case, beta strand, and tertiary structure and quaternary structure. We studied about the various forces that are involved, such as the van der Waal interaction, London forces, hydrogen bonds, hydrogen bond example in case of the pairing between Watson and Crick. And there were two types of pairing. One is the Watson-Crick base pairing and the non-Watson base pairing. This is the alpha helix. Then we came to thermodynamics, in which we studied about the Gibbs free energy, enthalpy, and entropy of the reaction. So we calculated this form. We understood how this formula works. Delta G is equals to delta H minus T delta S, and delta H and delta S are equally important, where energy minimization and entropy maximization plays a part in determining the position of the equilibrium. So this is the Gibbs free energy example in the real life. Next, we came to the application of Gibbs free energy to protein and DNA stability. This is the hydrophobic effect. So guys, thank you for listening today's session. I hope you enjoyed the session and you understood various concepts. If you have any doubt, you can post your doubts in the comment box, and I will be very happy to answer them. Please post your queries, guys. Please post your queries. I'm waiting for your reply. Please post your queries. All those who are present, please say hello. If you understood today's lecture, how was today's lecture, please type in the comment box and let me know. How was today's lecture? How did you find today's classes? Were they helpful? What all improvement should we make further? Please type in the comment box. Please type in the comment box if you have any queries or any doubt. Yes, guys, are you all not present? If you are present, then please post your comments in the comment box and let me understand that how you're getting the classes. Are the classes useful? Please post your comments on the comment box. Yes, Shraddha, how were the classes? Did you understand the concepts? Or were they useful? So guys, in the end, again, I would like to tell that please like. I think there are not many individuals who are present over here. So thank you guys to all of you who were attending the session today. And I would like to say that please like and share and subscribe to this channel and share as much as possible because you need to spread because it is a time for new uh, revolution in study here on Unacademy where you will be getting exclusive guidance from various top educators in the individual course category. You can use my referral code HY10 whereby you will be getting 10% off on your next Unacademy subscription. So thank you guys, thank you everyone. I'm here still. If you have any doubts, you can post your queries. I'm done from my side for today. Please post your queries if you have any doubt. I'm still waiting.